All 3.2 billion base pairs that comprise the human genome were sequenced in April 2003. Now, armed with this complete genetic code, the interactions between the cellular components that this DNA codes for can be studied. To better understand the interactions within a cell, scientists have turned to interactomes.
Speaking of the human genome, we know that DNA codes for cellular components of the genetic code, which interact with each other. But have you ever wondered, how? How does this process occur? How can we track the molecular mechanisms which govern these interactions? This can be explained through interactomes. Interactomes are defined as the complete set of molecular interactions that occur within a particular cell, whether that be physical interactions between molecules like proteins, enzymes, or metabolites. Interactomes can also tell us about the indirect interactions among various genes. Generally, interactome maps can be useful in determining protein or gene functions to further develop treatments for specific diseases.
Interactomes simplify complex molecular interactions within a cell using components called nodes and edges. Nodes represent the metabolite and macromolecule entities, including gene sequences, RNA sequences, and proteins, whereas edges in turn reflect the diverse interactions between these nodes, and thus can be functional and indirect, or physical and direct in nature. The two predominant methods that are used to identify these interactions and explore them include a form of screening known as high-throughput screening that allows us to efficiently analyze and test for specific biological activity, as well as mass spectrometry techniques through which we can analyze complex interactions, and particularly those that are protein-based.
So there are 3 types of interactome networks. All 3 use large datasets to visualize related genes interacting at their respective levels. So in the 3 diagrams shown above, the cell type and labels are not important. I'm showing them just to illustrate how these network maps can be used to represent different levels of cellular interactions, and an example of how these network maps are read will be seen later in the video. So firstly are protein-protein networks. These depict the physical interactions between proteins. Next are metabolic networks. These depict all biochemical reactions for a particular cell. In this diagram, the colored nodes represent enzymes interacting with their substrates. And lastly are gene regulatory networks. So typically these involve transcription factors binding to regulatory elements.
Interactomes can also be applied to practical, real-life studies of protein function and disease. Shown here is a complete protein-protein interactome of schizophrenia. Let's take a closer look. In this map, blue nodes and edges represent all of the known proteins and interactions, whereas red represents all of the new proteins and interactions found during this study. The functions of these novel proteins can be predicted based on their location in the network relative to those already known. This is called the neighbor-derived annotation method, which is based on the assumption that proteins with similar functions lie in close proximity to one another in a network. Interactome and network-based research is also highly relevant in the context of disease, particularly in the face of the ongoing coronavirus pandemic. In a recent study, interactomes were constructed to analyze the biological interactions between the coronavirus spike protein and the human proteome. This resulted in the following protein-protein interactome for SARS-CoV. The interactions here have revealed innate immune pathway proteins, which have been linked to much of the inflammation and pathology associated with coronavirus. Thus, new treatment methods, prevention strategies, and biological targets can be investigated, all thanks to interactomes.
Now that we've described what an interactome is and applied it to 2 real-life examples, the next question is to ask, why are they important? Well, 1, interactomes are crucial in discovering the function of unknown proteins. How? Well, we'd explore its interactions with proteins that have known functions. 2, Studying these networks allows us to identify genotype-phenotype relationships, which can tell us how the expression of a gene is affected. A disruption in the network can predict diseases, phenotypic changes, and the gain or loss of function in an organism. Interactomes can even allow us to identify gene candidates for disease. Overall, studying interactomes can help us to decipher the cellular and molecular basis of disease.